Hello, I'm James Lee, President of S-Vision LLC, formerly DR Vision Technologies LLC. It's my pleasure to highlight our poster, Intelligent Connectomic Analysis Tool for Dense Neuron Circuits. Intelligent Connectomic Analysis is a deep learning powered tool of Aiva for rapid boundary segmentation and cell reconstruction from EM volumes. The IKCA enables investigators to correlate structural with functional connectivity of neuron circuits. The IKCA processing pipeline steps, including image alignment and stitching, followed by boundary detection, neuron tracing, and the CLEM registration. The EM image acquired as a grid of tiles at each Z slice. The tiles are first stitched in each Z slice. This is an example of image. Each Z slice has 4x4 tile. Each tile is AK by AK, representing 40 micron by 40 micron. And then after the Z stitching each Z slice, and there will be a line along the Z direction to create a 3D stitched volume. The boundary detection is based on enhanced 3D UNet. It first generates a confidence map of neuron membrane, and then the map is threshold and then refined with morphological outcomes. This shows some examples. The neuron tracing is performed through a few steps. First step, connect small objects along the Z slice, then connect the large objects, and then conditionally merge small and large objects, and finally perform a 3D connected component. The CLEM including first draw the center line of EM image, then identify 17 landmarks at the branching points, and then find 17 corresponding landmarks from the LM image. Then using the landmark pairs to determine the transformation matrix. This is just showing some example of this registration. We verify the results using the standard matrix, the random score, and the information theoretic score. And also we use the adaptive RAN arrow for the neuron tracing measurement. This just shows some results. In B, we can show a perfectly reconstruct neuron, and the C and D have some errors in the reconstruction, mostly in the narrow portion of the structure. And then the corrective transformation is evaluated by F1 score. Here is an example of two LM fluorescence labels, and then overlay with the EM. So we find out when the two label overlap is a site of a riven synaptic sites, confirmed by EM. In summary, we confirm that the Aiva IKCA tool can accurately detect and trace neuron on EM volumes and can map, register, and correlate between EM and LM images. In the next phase, we would develop the complete IKCA tool, including deep learning powered synaptic connection, and validate the tool by applying to discover known and unknown neuron circuits and cell types. And our team, including Rachel Wang and Wanqing Yu from University of Washington, and myself, Luciano Lucas, Chi-Chao Huang, Hideki Sasaki, and Gaoyu Xiao from Aiva team, and also Grace Watts and David Swagart from Northwestern University provided the sample for the study, and then the project is partially funded by 3 NIH grants. Thank you very much. Please go to our poster for more detailed information.